So the next speaker is Albert Mondragon, who will talk about lysosome activity controls nurse cell death non-autonomously.
Great. Oops. So thank you for having me here tonight to talk about my work. I'm Albert Mondragon from the McCall Lab at Boston University, and I'll be talking to you about the role of lysosomes in nurse cell death. So we know a lot about cell death, right? We probably know a lot about apoptosis, characterized by cell shrinkage, the use of caspases, a lot about autophagic cell death or self-eating, the use of lysosomal machinery, and a lot about necrosis, where cells and organelles swell, right? But the important thing to note is that these three types of cell death aren't the only types of cell death out there, and there are a lot of other types of cell death that are currently being investigated. So in the McCall Lab, we study cell death in Drosophila ovary, and each ovary is composed of about 15 ovarioles, and these are progressively developing egg chambers from the anterior to the posterior side. So in each egg chamber, there are 15 nerve cells. So there's 15 nerve cells here that are connected to the oocyte through ring canals. And these nerve cells generate protein, organelles, and RNA for the oocyte throughout all of oogenesis. There are follicle cells that completely surround the egg chamber, all around the egg chamber. And there are two time points where cell death occurs that we study. The first is starvation-induced cell death, and this occurs at stage 8. And for more information on this, please see Sandy Cerize right here at poster D1062. But for the remainder of this talk, I'll be talking about the developmental program cell death that occurs between stages 10 and 14. So between stages 10 and 12, the nerve cells will dump all of their cytoplasmic contents into the oocyte. And between stages 12 and 14, the nerve cells become acidified, their DNA will become fragmented, and they'll be completely cleared by stage 14. So here's just another way to look at that data. At the bottom, we can see a stage 11 with 15 nerve cells, and by stage 14, all 15 nerve cells have been cleared. And so the graph is showing that in a control stage 14 egg chamber, there are no persisting nuclei. They've all been cleared. So the Cooley lab and our lab have both shown that apoptosis is not required for this type of cell death. So when apoptosis is blocked, these cells die normally. Our lab has also shown that the combined inhibition of both apoptosis and autophagy does not stop these cells from dying, right? So blocking these major cell death pathways, and these cells continue to die. So this suggests that there's another cell death mechanism out there that's removing these nerve cells. Could it be murder? So let's take a closer look at these stage 10 egg chambers. All right, so here are the nerve cells. The blue is DAPI. Here's the oocyte down here, and all along the outside are follicle cells. And so on the anterior side, there is a specialized subset of follicle cells called stretch follicle cells. And by stage 11, these stretch follicle cells begin to stretch and invade around these nerve cells. By stage 12, they've completely surrounded and encompassed these nerve cells. And by 13, you can begin to see that these nerve cells are starting to condense and look like they're dying. And by stage 14, all of the nerve cells have been completely cleared, and we just have really nice dorsal appendages. So a previous graduate student, Allison Timmons, has shown that phagocytic machinery is required for the removal of nerve cells. So this type of cell death is called phagoptosis. Phagoptosis was coined in 2012 by Guy Brown. Essentially what it means is that phagocytic machinery is required for cell death. And so what Allison showed is that SED12 junk and Draper, acting through and other phagocytic machinery, is required for the death and clearance of these nerve cells. Importantly, what we showed is that Draper is required for the DNA fragmentation. But we still weren't sure how the stretch follicle cells were actually killing the nerve cells, and so we decided to take a closer look at lysosomes. And so here we're looking at deep orange, or VPS18. Here's another way of graphing this data. In wild type, at biostage 14, all of the nerve cells have been cleared. So if we look at this kind of like a heat map, we can see that in the wild type, there's about 0 persisting nuclei. All of the nerve cells have been cleared. However, in deep orange hypomorphs, we can see about 50% of egg chambers collected had a severe persisting nuclei phenotype. At least 7 to 9 persisting nerve cell nuclei were in these egg chambers at a stage 14. We made DOR4 germline clones and showed that deep orange wasn't necessary in the germline or the nerve cells. It was required in the follicle cells, and we showed that whenever we knocked down deep orange in just the follicle cells here. Right, and so another way we can look at lysosomes or acidic vesicles is with lysotracker and acidophilic dye. So here at stage 11, we don't see very much lysotracker activity. However, at stage 12, we begin to see these puncta within the Streich follicle cells completely surrounding the nerve cells. And by stage 13, we can see that the nerve cells have completely become acidified. And of course, by stage 14, they've been cleared. All right, so here's just some live imaging of that happening. If we look at this spot here, this is a nerve cell. The nerve cells aren't marked with anything in here, and it's slowly being acidified. The green is the follicle cell membrane, and then the red is lysotracker. And we can see a lot of lysotracker puncta surrounding this nerve cell as it slowly becomes acidified and will eventually progress all the way up until this stage. Right, and so coming into this project, I really had 3 questions. I wanted to know whether lysosome, trafficking, and endosome genes were required for the clearance, and so for today I'm going to be talking about spinster. I wanted to know if lysosome function was required, so I looked at VHA16-1, it's a subunit of the VE-TPase complex, and I wanted to know whether cathepsins or lysosomal proteases were required for the breakdown of nerve cells, and I looked at CP1 or cathepsin L. So here we can see in wild type, again, there's not very many persisting nuclei, about 0 persisting nuclei in these egg chambers. However, in Draper, whenever we don't have the engulfment receptor, about 80% of those egg chambers have severe persisting nuclei. Now, when we start to look at the lysosome genes, here knocking down just spinster within the follicle cells shows that about 50% of the nerve cells persist in a stage 14. We can look at VHA16-1 and see that there's also about 70% of egg chambers that persist whenever VHA16-1 is knocked down. And we see something similar whenever cathepsin L or CP1 is also knocked down in the follicle cells. Right, so this shows that these lysosome genes are required for this nerve cell death and clearance. And so we can take a closer look by looking at the lysotracker. So to look at lysotracker, we're going to compare wild-type stage 13 egg chambers. This is when nerve cells usually become acidified. And we can see that about 70% of nerve cells in a stage 13 are acidified. And here in a Draper mutant, so this is Draper null egg chambers, we see a stage 14, and there was no acidification that occurred in any of these persisting nuclei, right? Very, very low percentage. When we look at the rest of the mutants, we can see the same thing with spinster. Lysosomes and lysosome activity isn't able to acidify the nerve cells. Whenever VHA16-1 or the VE-TPase complex is interrupted, we see a decrease in the amount of acidification in the egg chambers. However, whenever we knock down CP1 in the follicle cells, we don't see an interruption in the acidification. And so what this shows is that CP1 is required for the breakdown, not the acidification, while these other genes are required for the actual acidification of these nerve cells. Right, and so this is a egg chamber with spinster knocked down in the follicle cells, and we can see a lot of lysotracker puncta completely surrounding the nerve cells, right? But this lysotracker puncta isn't quite able to get into the nerve cells, and it's not able to acidify those nerve cells. And we see that an increase in size. These lysotracker vesicles are a lot bigger than usual, and there's an increase in number. And so this stage 13 egg chamber, though, is from a Draper null fly, and we see a decrease in the amount of lysotracker puncta and a decrease in the size. And so what Christina McPhee from Eric Bereke's lab showed a few years back was that Draper is actually required for autophagic cell death, and that you could rescue that death by overexpressing ATG1. So it's possible that Draper is actually has some sort of role in this lysosomal biogenesis, and it's really interesting to us that in this case, Draper's role might not be so much for engulfment and phagocytosis, but actually for upregulating this lysosomal machinery needed to acidify the nerve cells. Right, so what I've shown you here today is that spinster and VHA16-1 are both required for the actual acidification of the nerve cells. I've shown you that CP1, or cathepsin L, is required for the breakdown and removal of the nerve cells, and this work suggests that Draper might actually have a role in upregulating lysosomal machinery that's needed for the actual acidification process rather than engulfment. And with that, I'd like to thank everybody in the McCall Lab, specifically Ala Netskaya, Anthony Ortega, Wanheng Zhang, and Allison Timmons, the Fly community, and the NIH. Thanks.
Hi, I'm curious if you know whether this delivery of lysosomes by the SFC starts exclusively after dumping is complete. How is this prevented from acidifying the oocyte through the ring canals?
Right, so it definitely happens after dumping is complete. The delivery mechanism, we aren't sure, right? So we know that the Streich follicle cells are murdering, and we know the weapon is lysosomes, but we don't know how those lysosomes are getting there. So we don't think that it's exocytosis that's occurring. It's really strange. All of our studies looking at lysosome secretion and exocytosis haven't really shown us much. And if we look at the live imaging, we can see those large vesicles almost look like they're kissing the membrane of the nerve cells and just bouncing off, right? It looks like kiss and run fusion, I guess. But we're still working on how it's being delivered. Thanks.
So I'm just curious, what's causing the massive formation of these lysosomes in the Streich follicle cells? Do you think it's a timer, or do you think there's some signaling that's happening to induce the formation?
For the lysosomal biogenesis, right. So I think it has to do with MITF. So it's a lysosomal biogenesis transcription factor. So when this is active, you get a lot of lysosomal biogenesis. And I think I have some preliminary data suggesting that that's involved in the process. But I'm looking into it, trying to figure that out.
Hi, I'm a little bit confused about the acidification process because at the last 2 slides you mentioned that the nerve cells don't actually become acidified, but there is some nuclear breakdown. So what is the actual role of this acidification step?
Right, so I don't think we've ever been able to show any mutants that are acidified or that are not acidified but have DNA degradation. So what I was saying was that acidification is blocked in some mutants like the spinster, the Draper, and the VHA 16-1, but acidification was not blocked in CP1. In that case, it seems like cathepsin L from the Streich follicle cells is somehow being delivered to the nerve cells and is necessary for the breakdown and the breakdown of the nucleus after it's acidified.
Okay, thank you.
Yeah, thank you.
One quick question. I was wondering if your lysotracker positive structures are really lysosomes, or if they could be late endosomes, and then you could have MVB exosome secretion for acidification.
So they're gigantic, and I think lysosomes are only supposed to be about 0.1 to 1 microns, I believe, and those structures are much, much larger. So I'm not sure what they are. I've tried LAMP1 staining, and it hasn't worked out for me. I'm not sure if it didn't work out, or if it wasn't, or if these just weren't LAMP1 positive. I'm trying to identify those right now. Yeah, that's a good question. Thank you.
Thank you.